Hello everyone, welcome to MMBS111 Introductory Microbiology. This is the final mini lecture for Chapter 8, 8.6. We're going to be talking about microbial genetics and genetic engineering. There are three learning outcomes for this mini lecture. First, explain the importance of restriction endonucleases to genetic engineering. 2, list the steps in the polymerase chain reaction, or PCR. And third, describe how genes are cloned in bacteria. All right, so restriction endonucleases are also known as restriction enzymes, and these are enzymes that their normal function within bacteria is to break the bonds of nucleotides within fragments or pieces of foreign DNA that ends up in the bacterium. It protects the bacteria from DNA that is incompatible with its own chromosome and recognizes the DNA of bacteriophages. It has been used by us in the laboratory to cut DNA at very specific desired locations. It is an absolutely necessary component of DNA technology. Without it, we would not be able to clone genes or produce some of the recombinant products that we are currently doing. The restriction endonucleases cut the DNA in staggered symmetrical cuts that we call sticky ends. And these sticky ends help to connect new sections of DNA that were also cut in the same way. They typically recognize 4 to 5 bases within the DNA sequence, and then they cut at those specific sites and leave sections to overhang so that new sections can be added in. So you can see here the action of restriction enzymes. So they're kind of like scissors, right? They're going to cut the DNA in a very specific location, and we call these palindromes. They're sequences that are read the same on the 5' to 3' end as they are on the 3' to 5' end. So it's going to recognize those very specific sequences within the DNA, and each restriction enzyme recognizes a very specific sequence, and it cuts in that very specific location. So we've identified actually a number of restriction enzymes that will cut DNA in different places. Once that DNA has been cut, then we can combine DNA from different organisms that have been cut with that same restriction enzyme and combine those two different DNA fragments. We have also developed technology using restriction enzymes and some other tools to analyze DNA to help us determine things like size and particular genes. Gel electrophoresis is one tool that has been developed that we use to analyze DNA, and we use restriction enzymes in this process to cut the DNA into multiple fragments, and then the fragments are added to a gel. And since DNA is negatively charged, when we add an electric current to this gel, the DNA is going to move towards the positive pole in the gel. And the larger pieces of DNA are going to move at a slower rate than the smaller pieces. So generally, a fluorescent dye is added to the DNA so that we can then look at those gels and determine which fragments based on their size. So this helps us to kind of create sort of a genetic fingerprint. Usually a marker is added to the gel, and that marker helps us to know what the size of each fragment is, so we can compare the DNA sample fragments to the known marker fragments, and that helps us to determine the size of each fragment. PCR is a tool that allows us to magnify the amount of DNA that we have. It's sort of like a Xerox machine. It makes multiple copies of the same fragment. It requires the use of some primers that are recognized so that we know the polymerase knows where to amplify or where to copy the DNA. We also use a DNA polymerase that was isolated from a thermophilic bacteria, because the PCR process uses some pretty high temperatures in order to separate the two DNA strands, and the DNA polymerase in the thermophilic bacteria is able to survive those high temperatures. And then we use a machine called a thermal cycler, and it cycles through a sequence of temperatures automatically, so that this process just happens repeatedly automatically. So we go from those temperature changes that are needed to separate the DNA strands and then reanneal or rejoin those DNA strands once the replication is finished. So this image just shows you the general, the basic stages of PCR. So the first step is denaturation, and this happens at really high temperatures, 94 to 96 degrees Celsius. So this is why we need that thermophilic bacteria DNA polymerase, because other DNA polymerase enzymes are going to be denatured at this temperature. So during the denaturation stage, we just want to separate the two strands of DNA from one another. The second stage is annealing, and this is where we are adding bases. So we add the primer. That primer lets the DNA polymerase know, hey, this is the part that you need to replicate, and then bases are added during the elongation phase. So we have annealing and then the elongation phase. And then this cycle repeats. So if we start with one double-stranded fragment, we can end up with multiple copies of that fragment in a very short period of time. 64 copies after just 6 cycles. So this allows us to replicate desired gene sequences in a very quick amount of time. So the primary intent of recombinant DNA technology is to intentionally remove the genetic material from one organism and combine it with that of a different organism. And through this process, we have been able to mass produce things like hormones, enzymes, and vaccines for medical use. This graphic here shows you the steps involved in cloning a gene. So when we clone a gene, we first want to identify the gene of interest, and that's usually going to be a gene that is found in an animal or a plant. Sometimes it's a gene from a microorganism, and we want to replicate that gene's products in a bacterium. The bacterium are typically the vectors. Sometimes we use yeasts because they reproduce quickly. So we're going to get the products from the expression of that gene, so the phenotype, quickly and in large amounts. So we're going to cut that gene out of the host or donor organism, and we're going to insert it into a plasmid or into the chromosome of the cloning host, which is typically a bacterium or sometimes a yeast. We're going to use restriction enzymes, the same restriction enzymes, to cut both pieces of DNA. This gives us matching sticky ends so that those two pieces of cut DNA will match up when we combine them. Then we're going to put that plasmid into a bacterium, and because bacterium multiply as such a quick rate, short generation times, we're going to get the product of that gene very quickly. And then we can harvest the product, whether it's a particular hormone or an enzyme, maybe it's insulin. We can harvest that product from that organism. And that is the end of mini lecture 8.6. So here are some questions to check in. Make sure that you understand the content from this section of the chapter. So go through these questions, make sure you can answer them correctly. And here is our critical thinking question. So in 1977, scientists discovered a way to force bacterial cells to produce human insulin, which could then be harvested, purified, and used to treat human diabetes. Explain the process used to clone the human insulin gene. So think back what we just talked about. Why is insulin produced through recombinant DNA technology safer than insulin harvested from an animal source? Okay, so think about these questions. How would you go about answering these? These are some of the types of questions that you're going to encounter on the exam, so make sure you go through these. Also, go back to the learning objectives. Make sure that you understand the concepts there. Remember that you can watch this video as many times as you need, and hopefully it was helpful.